the first lecture about secondary structure prediction today. Before we go, announcements. By now you all know there will be no lecture May 29th, June 3rd, June 10th, June 19th. Recap, secondary structure prediction. So first of all, again, the idea is that a protein sequence uniquely determines its structure. This is important because structure determines function. So you throw a stretch of amino acids into a solvent and they adopt a unique three-dimensional structure. This is the background for the prediction of structure and function, and this is the background for aligning sequences. Although there's an evolutionary relation between them, the biochemistry is an important part of the signal. We can use that signal for comparative modeling. We can model about a third of all the proteins, and that has been assessed. How far structure prediction can go has been assessed in CASP. The idea of secondary structure formation is that secondary structure is stabilized by hydrogen bonds. Why do we have secondary structure? Now, let's get back to that question. Why does secondary structure form? So there's hydrogen bonding that defines secondary structure. The hydrogen bonds form, and therefore you form secondary structure. But that's not the meaning of the issue that I'm addressing here, or the question that I'm asking. The question I'm asking is, and on the next slide you see one of these examples of a structure, and I've shown you many cartoons. The arrows here mean beta strands. Here is the beginning of an alpha helix. So when I showed you structures, you always very often saw essentially those two elements. You saw strands, arrows and the like, alpha helices, and something in between that I, in the last lecture on Tuesday, referred to as the non-regular state, and therefore I call it other. Sometimes people refer to it as loops. Bless you. But why do these helices form? Again, yes, true, they're stabilized by hydrogen bonds. Why do you see them every single time? Because you have so many hydrogen bonds? Hydrogen bonds could do other things. Why do they just do those things? So part of the second question I'm asking, why do they just do those things, brings us back to what I talked about on Tuesday, when Linus Pauling published many papers in PNAS that essentially were many different things that could form. And to some degree, we still don't know why the others actually don't form, why they're not observed. They would be feasible. Those, now we say, and today's computation of energies confirm this view, those two states that we see are the energetically most stable ones. So in a probability sense, something that has a good energy is most likely to form. So this is the reason why these two main segments form. But what do we learn from observing helices? On strands, what does it help us when we look at a structure? So the first thing that it helps us, and again I should have pulled up the example that I had in previous slides, where you see the per residue view, or the alpha chains, the C alpha residues, as opposed to these arrows. So the first statement is, you immediately see something. You can recognize, so when I show you this single atom view, you cannot recognize anything about that structure. You see a bunch of molecules, a bunch of spheres. In this view, you can begin to recognize themes. And these themes, it's not about recognizing folds, about following evolution. That is part of the game. But part of the game is that these themes also tell you about binding. These themes, this particular loop here may be important for binding, or this long one that winds around. You recognize this long loop only because you sort of see these arrows. You follow these arrows, you see the long loops bind. Binding is very often done in loopy regions, or in what I call other. So in the regions that are neither alpha helices nor beta strands. So the way to think about these alpha helices and beta strands really is like they provide stability. First of all, we believe there is a rapid event, that there's a rapid, so you have the stretch out chain, you throw it in the solvent, and there are sort of breaking points. somebody left a nice protein chain here. So the breaking points are, just imagine if this is the protein, you throw it into solvent, and then immediately well, with a hose, you can do the same thing, or with this thing. You immediately have 1, 2, 3 breaking points here, and they lead to a rapid collapse. That is good because there's a lot of hydrophobic residues in the molecule, and they want to stick together. They want to get away from the water. Hydrophobic means they don't like water. They're not friends of water, as opposed to hydrophilic. So you immediately have a rapid collapse of the molecule, but it already gives you a better energy. And then supposedly folding happens there, and then you in fact fold into helices and the like. Bless you. So the helices in the next step help you to keep up with the rapid compression of the stretched out chain. That's the idea we have today. That's the idea we can observe when we can simulate folding in, for instance, the machine Anton that I talked about. But mostly function, again, the helices and the beta strands, they're very important for function, for the structure formation, for folding. They tend to be just backbone or just the pillars upon which the buildings stand, or the part of the building stand that does the function. Well, I don't know, imagine that the loop in a protein chain here is like a door. So there's nothing about a building without the door, right? At least for you to use that building. And in some sense it's similar here. Okay, let's talk about secondary structure prediction today. And the first issue here is about words. Secondary structure prediction. Why is it called secondary structure? Yes, this is biology. Somebody sees something and comes up with a word. So you can...
Because we get rid of something, so it's not the...
that's a great idea. Biologists, you're too mathematical. Biologists don't think in terms of information content, or old-fashioned biologists. So today, biology is in equal parts, I would say, in some sense, computational biology and experimental biology. And computational biologists think in terms of information. But the traditional experimental structural biologists, they are more visual people. So throwing away an information piece, and I'll get back to another aspect in a sec on that. Why could it be secondary structure? Somebody?
It's the next step after the sequence?
Yes. Well, yes, you're totally right. Have you read something about primary structure? So historically people refer to the sequence as the primary structure. It has no structure. So the world of biology is full of words that are not totally intuitive. But yes, exactly that is the reason. So primary, the sequence was considered the primary structure. And then came the secondary structure, sort of the next best thing. And in some sense, clearly, the way we look at it, or I look at it, as an information content is sort of a reduction from 3D, which is the next step. But that led to many people considering secondary structure as 2D. Secondary, right? That's sort of another misleading intuition. Secondary structure, again, I put up this slide here, is a 1D string. Information content, this is 1D. Absolutely not 2D. 2D is a contact map. Has nothing to do with the secondary structure. The connections between the secondary structure here, how the strands form a sheet, that is contacts. That is 2D. But the mer placement, which residues in a helix, which residues in a beta strand, is primary structure. Again, for most of the proteins we cannot model 3D structure, so we have to simplify secondary structure. Let's get back to DSSP. I actually omitted on Tuesday the pi helix here. There are 3 types of helices, 2 types of strands, and the other 3 states are considered other, non-structured. Now, the first step after you have this classification using DSSP, using structures to classify proteins automatically from 3D structures into assignments of secondary structure, you can actually analyze structures. And the first thing that the authors of DSSP found out, captions under, was that some peptides, so some stretches of amino acids, typically referred to as peptides, are always observed, or were always observed, so this paper was 85, were always observed at the time in alpha helix, others in beta strand, yet others in this other state, and some were seen by the authors. Those are pentapeptides, 5 residues long, were observed both in alpha helix and beta strand. Okay, how could that be? How can you picture that? You have 5 residues that are seen in 2 different 3D substructures, secondary structures, helix and strand. I've shown you pictures of proteins, and by now I bet you have seen enough to see that they really look different. Helices look like that. Beta strands are extended structures that come together, very much in the way I folded this chain here. So beta strands are things that are like that. It's nothing like an alpha helix. How can these pentapeptides be observed? Yeah?
Alpha helix and beta strand are both made because of hydrogen bonds. So it's not that surprising that we have the same source for both structures, and maybe we can imagine that this 3 sequence gives just something in common for the interaction in common of both, whereas that the other part of the group for helix had some other.
Let me become emotional. This is one of those moments. I actually lift my hat for you. Thank you. You did my day. This is a great statement. Yes, exactly. And I'm so impressed because this means that there is something that I got across that led to deep understanding, and that was a wonderful moment for me today. Thanks. Yes. That's exactly it. So you have the same origin, so you have this, the difference between alpha helices and beta strands is exactly in the way these bonds are formed, and that has to do with what is next to you. So you could go up and keep going into helix, or you could happen to get next to a beta strand, and here I should show a beta strand now, and suddenly it zips like a zipper. Okay? By the way, there's another motif that is really zipper-like, and it's called a leucine zipper, where 2 helices come together like that. So again, hydrogen bond formation, hydrogen bonds are very weak, and typically they will not bind something like 2 helices. Beta strands have every second beta strand, so there is a sort of stronger connection. Alpha helices, they can maximally bind if 2 alpha helices come together with every 4th residue. So for those to zip up, it needs a little bit more things to fit, and those are leucine zippers, and this is in fact the way you wind up helices around each other. And is anybody in the room who has used these simple drills where you have in fact 2 intertwined pieces of metal, and then you have something that walks around this, and you can drill with it, so you move it up and down and drill with it. So you have seen something like that. So these things, those are called helices, and they in fact drill holes into cells, exactly the same way as this tool that you have seen. So where you move around this, and by moving around you create a force that makes this intertwined coil, these helices become a drill that can punch a hole into something. ATPase is an example where that really exists. And that's one of the things that fascinates me about proteins. They are really simple machines, and you see a lot of what you see in everyday life realized in these protein machineries. Okay, so we can understand that there is some similarity. And our goal is look at the sequence, the primary structure, and predict secondary structure. So which residue is in a helix? And the first paper was from St. Georgi that predicted proline to break alpha helices. Okay? That paper was in 57. Does anybody have to say anything to that? From what I presented to you? Remember on Tuesday, I told you when the first structures were published. Kendrew and Perutz. So when you read these numbers here, that's before the observation of structure. Again, we have an example where we have the first secondary structure prediction method published before they ever had seen secondary structure, which is one of those remarkable points in history, clearly visionary. The idea was that proline stands out from all the other 20 amino acids, and that in fact completely changes the way that the backbone is traced. So it creates a kink in the backbone, so to speak. And this kink, was the argument here, would break a helix. If the kink breaks a helix, then the proline would be the last residue in a helix. And that would be a way to sort of predict the end of a helix. That was the rationale. Now, how can we use this idea to develop a prediction method that does a little bit more than proline? What's the simplest prediction method, secondary structure prediction method that you could possibly imagine? Somehow along the lines of this idea here of the proline. Yeah?
Well, you just find some markers like proline here.
Yeah.
And then you just check where's proline, and you say, yeah, there's proline alpha helix before that, and then you.
So the way you imagine that, let me just repeat what you say. Is good practice to always repeat statements and questions when you are a speaker, but maybe people in the back of the room didn't hear that. So it is a good idea. His idea is to repeat the idea of the proline and find other markers, as he calls it. So motifs, as I would tend to call that are observed in particular secondary structure states, and then do the same thing that is done here for proline for other residues or for other amino acids. True. That's one way in which you could do it. Any other way? In some sense much simpler, avoiding the word marker or motif. What's the simplest thing that you can do? Does that give it away, the last bullet point? What I mean? Anybody? Odds? Yeah?
Yeah, you just do it statistically. You just say that if there's a proline here or if there's a cysteine here, then in all the proteins that we know that we have observed this and this, in 30% of the proteins this happens, in 15% of the proteins this happens, and then you just assign odds, and then you see which odds work out the best.
Yes, exactly. So you simply go over in this particular case you have two states of secondary structure E and L. Don't know why I dropped the helix. Oh, because in this particular case, that's just part of it, whatever. In this particular case, there's only beta strand. So this is your observation. Blue is the secondary structure assignment from DSSP. The dot here means that most likely is a strand, the whole thing, which is not really identified automatically. Detail. Every single residue, so where you have a blank, you have the state other. You simply count. Q is observed in other, that gives a count for L, shown L here, of 1 for the Q. I'm sorry. And we continue going, right? You do this for all residues and for all structures that you observe. And you get the simple statistics, and the simple statistics is used to, when you see that particular residue, you predict secondary structure at a certain probability, and at every single position, you use the probability of what you expect. That is the idea published first by Chow-Fassman. 57, again, it dates back to St. Georgi, but the Chow-Fassman really was worked out in more detail. Chow-Fassman 74, Barry Robson in 71, and Gore, Garnier, Oscar Thorpe, and Robson, which is another of these great teams. Garnier, in this particular case, was the biologist. Robson was the brilliant mathematician, statistician, and Oscar Thorpe sort of could do. It was a biophysicist who knew about theory, biology, and algorithms, so he somehow connected the two worlds. That's 78. And the principle is this idea. Is this idea worked out over a decade? Where do we get secondary structure from? Well, I showed you DSSP. But initially, DSSP, remember, I showed you the time here, 78. You may remember that DSSP was published in 83. So DSSP doesn't exist at this point. Where do you get it from? There is no algorithm. So we're talking, again, the first methods here published 74, 71, the last in this first class, 78, all predates any attempt to automatically classify structures, 3D coordinates, and spit out by a program their secondary structure. So how can you get secondary structure?
By manually looking at them.
Exactly, by looking at it. And that, in fact, is what people did. So they classified whatever structures there were. The number was not that high, so it was not that much work. And essentially assigned secondary structure according to what they saw in 3D. So to the images that I have showed you of these C-alpha traces may be confusing to you, but if you can certainly imagine that if you stared at it for many, many days, you would begin to make sense of it. And that's exactly the way they did it. Now, how would you then assess the performance? What would you look at? What would be your criterion for having a good secondary structure prediction method? What is the measure? Yeah?
Well, you can do a direct match, and you say at residue 15 in the expert assessment, here the alpha index starts, at residue 30 it stops, and then we check whether it matches pairwise the assigned secondary structure, how well it matches.
So I'm not entirely sure, so I'm not going to repeat that statement now. I'm not entirely sure what you mean. So do you mean that you match helices? So you count one helix is right, one beta strand is right? Is that the way you say it?
We had in the DSSP.
Oh, again, we are before DSSP. But it will be the same for DSSP, so keep going.
Yeah, you have those for this residue is part of the helix, and then you check the automatic method, and you check if the automatic method also says this residue is part of an alpha helix.
So my question for you, the measure that you are inventing here, are you counting one strand, two strands, and you match them or you don't match them?
No, I'm assigning. What I do, I take an expert crystallographer, I tell him...
Yeah, I know you like to grab your own experimental biologists. These guys come expensive. They don't do that. But this is not what I mean. This is just a side comment. So say in this particular case here, I roughly do what I have on the whiteboard up there. So say my prediction would be this. Let's leave the experimental people out for the time being. And my question really is from what you see on the whiteboard. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, simply because it's easier to divide by 20 or whatever, to divide by something. So, let's get back to what you're suggesting. What would you count?
Well, I counted 20 and I count 1, 2, 3.
Oh, so you count individual residues. So you ask how often do you match a particular residue in one of those three states? Is that what you say? Any other idea? That is exactly one way. In fact, it's referred to often as Q3, and the way that makes the most sense is by saying this is a three-state accuracy. So the percentage of residues that are correctly predicted in those three states. So helix is not here, but the dot is an L, Q, E, and H for helix. What else could you do?
we break up a secondary structure or we join a secondary structure.
Exactly. So you could look at, let's just extract from what you say what I would call the main part of the idea, which is to look at segments rather than individual residues. So rather than having individual residue being right or wrong, say I catch the helix, or in this particular case, the beta strand. So you said whether you break up, so back to the proline idea, whether I break up a helix and whether I find that the helix is broken up. Now in this particular case, so the way I constructed this example, is that beta strand predicted correctly? So this has 0 overlap. What about this one? How would you handle that question? So clearly we need something, some way to measure the performance. The simplest one is Q3. Most of what I will talk about, I believe all I will talk about today is related to Q3, or almost all of it is related to Q3. But you also understand that ultimately we will have to ask a question that relates really to something on the level of segments. Do I catch the helices? And how would I respond to that question? How can you propose a solution? Let's think about the hack, yeah?
If we know that the major, all of the sequences are smaller than a certain amount of parts of this unit,
yeah.
We could say we can check all the subsequences which are smaller than this length. So to avoid to do all the subsequences, but I know that it will be very...
I'm not entirely clear, I understand your point. So are you talking, when you say subsequences, are you talking about helices? Are you going to look at the length of helices, or are you looking...
We could try to match...
so a line secondary structure?
Yeah, on these two...
Oh, be careful. We don't want to align secondary structure, because the secondary structure prediction method says for that particular position, it's an E.
Yes, but we can compare, so once we can do all the pairwise comparison, and
yeah.
we can compare sequences 2 by 2.
Yes. 2 by 2, you mean this pair?
No, so it will be two pairs. Like the first and second one, and the first and second one for the second protein. So I take the first two on the first structure, and I take the first two on the second.
These first two. The first, are you talking about the first two residues or the first two? Yeah.
First two residues, yes. And then I just check the second and the third one, and the second and third one.
So you move in windows of 2?
Yes, and then I move with windows of 3, and then 4.
Yes, but that idea brings us back to something that relates ultimately to this. You're essentially in some different way by having different scans. So now you replace this as an individual residue level, and now you replace it by a sort of region of, say, 5 residues. That is a different idea, but it's an idea that is independent on where the helices sit. Oh, what I didn't tell you, but you get an idea. No, you don't get an idea. But helices have very different lengths, and beta strands too. What we could do, maybe that could be at the base of what you say. We could ask for all the beta strands of 4, how many do I match entirely right? For the beta strands of 5, how many do? That would actually be a way to do it that I've never seen before. Maybe I would have to play with that. I believe, I'm not sure whether that's what you meant, but that could go somewhere where certainly I have never been. But what did you mean then?
Well, I think that the result is not really the same, but approximately, but it's, to try to catch all the sequences that are well predicted with a length of 4, and then of 5, and then of 6.
So that, again, that is essentially what I said before. In some sense, you match all the beta strands helices of a certain length, and to ask how many of those are completely right. And that is one way of doing it. That way of doing it assumes, or would work only if the answer to that question, how many of the beta strands with foo are right, is answered more than 0 times by methods correctly. So in this particular example that I put on the whiteboard, you have a secondary structure prediction that is actually, after all, by the Q3, not too bad. Right? By the Q3, you have 20 residues, and you make 4 mistakes. Is that true? 4? No, you make 6 mistakes. But still, most residues are correctly predicted. But by that method, we would have 0 that are correctly predicted. And in fact, I believe even better methods would pretty much get close to 0 in that. So one way is to sort of come up with hacks of saying, okay, it's not important that I have to really match the entire thing of foo, but as long as the overlap is half or some parameter, and you begin playing with parameters. That's one way of going, one hack way of going. Another one is you could sort of look at compiling percentages with this sort of allowed deviation. You give yourself 1 on either end. In this particular case, you would then call that right. If the E would be 3 times longer, so that's different from saying half overlaps. So these two would, the one that I showed you before and this, would be equally right by that. You somehow ignore ends. That would be a work, but it would violate what initially triggered your idea, which is in fact to see where helices and beta strands start and end. That would be totally violated here. But we begin to see that there is a little bit more we have to do. Okay, back to the Q3 is defined here, originally by Schulz and Schirmer. Now, using this tool, the secondary structure prediction for the first level methods, the Gore methods here, G-O-R, Robson, Osgothorpe, Garnier, Gore methods, was estimated to be 63 in this overall percentage accuracy. So how many of the residues are correctly predicted? When DSSP came along, what do you believe happened? So the authors published their visual assignment-based probability methods and said they would be 63% accurate. That was done in the 70s. In the 80s, suddenly somebody has an automated method, this one here, DSSP, and does it for 50 proteins. Previously they had done it for just less than 10, but in that ballpark. What will happen to those methods? What will happen? And it, in biology always happens somehow for prediction methods. So with that assessment, it turned out, actually not immediately, but later turned out that the prediction accuracy is in the ballpark of 50 to 55. And I just saw the other day that this is attributed in Wikipedia to that paper, which actually is not true and maybe goes back to something that I've been running around and saying. But this was actually established in '94. So let's move on. We can consider this simple probability idea as the first generation of methods. How can we extend that? What can we do to come about with a second generation? So that's the simplest thing you can do. For every single residue, you have a certain probability that it is in one of those 3 secondary structure states, and then you predict secondary structure according to the max of that. Is there anything you can do that is sort of more advanced? Still simple, so we're still far away from talking about machine learning. We will talk about machine learning today, but not yet. Something simpler. What else can you think about?
Maybe I don't take simple letters, but I take whole words and say, okay, the first letter was A, the other was B, so I have for B another probability for another structure if A equals before.
That is very much aligned with what you proposed before, yes. So that's the idea. In fact, I bring this up because sometimes history is important. So DSSP They developed their first method that in fact automatically assigns secondary structure for known structures, not because they felt this is an important thing to do. Turned out to be a very important thing to do, and it has been used by everybody ever since. And there's still no good improvement over that method. But ultimately what they wanted to do is derive a method, they call it segment 83, that in fact uses words. So compiles a probability for a word and looks at entire secondary structure segments. The problem was that they needed more data than 10 proteins for which they had the assignments, and they were too lazy to in fact do the visual assignment themselves. They both were people who felt it was okay to spend 2 years writing a computer program that does it, but not to spend 2 months and in fact doing it visually. And reality proved them right in the sense that it started for both their fame. The other part of the story, segment 83, the reason why this was set up, never got published. But DSSP made it. But anyway, so you have the first idea towards segments. You look at words here. Any other idea? How can you use probabilities? It's not quite another idea, but it's also not the word idea in that sense. So the word idea, now I call a word a secondary structure, so helix, or something that relates to helices. Any other idea in which we could move from a single residue probability to something a little bit more advanced? Not considering secondary structure at this point. Yes?
You can look for specifically spaced sequences of hydrogen.
You like motifs. I love in life how consistent people are to their way of thinking. Yes, that's a great idea. So now in the initial motif, in the first round we discussed the proline breaks and other motifs of that type. Now you say, let's not just look at an individual residue, let's look at 5 consecutive residues and form motifs by those. Great idea. And it's an attempt that works very well. There is no commonly used secondary structure prediction method that implemented that had been used, because people did not come up with that idea in time to use it for that. That idea was used for identifying functional motifs. And by the time it was applied to secondary structure motifs, other methods had become too good. But it's a great idea. Now, what about probability? Any idea that sort of uses the word probability? And more than single residues. Yeah?
Could look more at the probability of entering a helix and then continuing a helix.
That's true, the proline. But that essentially, in some sense, how would you do that? So far, we have probabilities for every single residue to be in a particular secondary structure. Now you want to redo that for beginnings and ends. Just recompile the statistics only for beginnings and ends. Is that what you say?
I would do it basically in sequence. I would start from the beginning with the probability of that being a helix, and then the next one.
Yes. But again, so what you say by going through the sequence from one end to the other, here is that a helix? Is that breaking a helix? Essentially, now you replace what I described before, Gore, which has a probability for every single residue to be in one of 3 secondary structure states. By now, let's call it, I don't know how you do it, but you could have 4 states, where the new 4 states would be, it starts or ends a beta strand, it starts or ends an alpha helix, and you assign a probability for that. Is that your idea?
I would rather say the probability of a given one to be a given peptide to be a helix, given that the previous one was a helix, most probably most likely.
So now, yes, I believe I recognize Gore 3 here. So essentially what you do now, and I believe that's what you meant, is what is known to the field as GOR3, again for those three authors, Garnier, Osgothorpe, and now I don't even show the paper where he's still on, and Robson. That's the third iteration of GOR methods. And essentially the idea is really that you compile the probability, and I don't know why I don't have the slide here, that you compile the probability, so given that this is a proline next to a leucine, next to an isoleucine on either end, what's the probability of that to break, start a helix, be in a particular secondary structure? So now I have conditional probabilities for segments. Okay? This is no longer, it's not the word idea. So we're not, in that sense, predicting a sum over the probability of the whole thing being a beta strand. The probability is still for an individual residue, but now it's conditioned upon what is before and after. Okay? So now you no longer look at individual residues, but you sort of add up the probability for some segments of, say, 5 consecutive residues. This type of method I like to refer to as the second generation method, and there are many, many, many of those, many publications. They reach levels of 55 to 60% accuracy. So we are reaching sort of the beginning of the 90s, the end of the 80s. We now have a bunch of structures. In fact, at this point we reach almost 100 structures, and people are throwing almost anything that they can think about, including machine learning, I'll get back to that later, at this problem. And essentially, they sort of hit a limit at around about 60%. The publications are a little bit higher. Again, these numbers here come from publications a little bit later. But there is some limit. So problem number one, we don't reach 100%. At the time, they argued that the level was 65%, and they argued that 65% is the max you can reach. How could that be? Why do you believe the argument would be cannot reach more than, in this particular case, with 65%?
Maybe the method is introducing errors. So
that is true. So I make a mistake of 35%, but the question is, why would people argue that there's an intrinsic problem in the method? Why you can never make less than 35%. So saying that the method makes errors, yes.
So the method is just sometimes they have a choice in the model, and they make a decision. That in the most case,
all you say is not a principal thing. You can say that about any number.
No, it's that the way that they're comparing, sometimes they're making choice to improve the average accuracy, but they just make the accuracy lower in the best case. And if we compare the same structure with this method, we will have 60% accuracy.
The point is, my criticism to your comment is, in principle, you can make this. So if I wanted to predict the size distribution in the room, okay, I have a simple Gaussian. I take it from the web what the distribution in some countries is, and I apply it, and I get a certain distribution, and then I look whether the distribution I have and the distribution I observe are similar. Okay, and what you say can explain the mistake. You make some mistakes, essentially, is what it boils down to. And mistakes may add up. Okay? But you're not saying anything that is specific to this problem. Yeah?
Because not all the information needed is in the sequence.
What could be missing? Oh, yes. So he says two things. So he says, not all the information that you need for this problem is in the sequence. Now, let's get back. That's the first statement. I omit the second statement. That was a little bit misleading. All the information is in the structure. We can assign secondary structure from the 3D structure. But so get back to that point. All the information is in the sequence. You're on the right track, but this, I'm going to get you onto ice now. I said that you take a sequence, you throw it into solvent, and it adopts a unique three-dimensional structure. The sequence knows this. So all the information is in the sequence.
You also mentioned that the environment...
Yes. Great. Well, wonderful. Thank you that you bring that point. There is more to it than the sequence. The environment is important. But again, and I also mentioned that what I describe as the Anfins and Epstein experiment, we don't know whether it's always valid. But we have some proteins for which this is valid. And where the environment, all we need from the environment, is water. That is not specific. So I take two different amino acid sequences and throw them both into water, into the same water, and I get different structures, and I get always the same different structures. So the way sigma structure is formed is in the sequence for those cases. That's absolutely clear. So you're on the right track, yet I got you onto the ice. The way you say it, it's wrong. How can you make it right? Or how can anybody else make it right? Remember what we put in here. Gore 3 uses segments.
The segments are not long enough?
Exactly. The segments are not long enough. The information is in the sequence, but these segments are in fact relatively limited. This may work for helices where the hydrogen bond formations are in I+4. So if I have 5 consecutive residues or 11 consecutive residues, in fact I see for 11 I see the hydrogen bond forming partner on either end if I predict the state of the central residue. That's easy. But for beta strands, you see the beta strand runs here, 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 and then comes back. But wherever the hydrogen formation is, that is not going to be visible. So the binding partner will not be visible in a segment of 5. This would immediately also suggest that in fact secondary structure prediction accuracy would be much lower for strand. In fact, these methods have only 40% accuracy for strand. So overall they reach something like 55 to 60, then they said it's 65. But for strand, it was almost random. So completely consistent with what we observe. The other problem was really that we have short segments. And let me illustrate that problem here. So that's the sequence, that's the observation, and that's a typical second generation secondary structure prediction method here. So you have E, H, 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 E. So the point is in terms of counting every single residue, this would add up to 55 to 60%, but you're not really, in terms of what you suggested, of counting the strands of length 4 or 6 that are entirely correctly predicted. This is 0. But not only that, for you as a user, or if you were a biologist, the question immediately would become, most likely this is a long helix, so cut the E. When you look at the reality here, you see that the E actually contains some information that was right. When you have ice here, how would you turn this one? How would you turn this one? What does it mean here that you have 2 strand residues? Just cut them both out? So the fact that you predict something that is much shorter than observed on average makes the prediction not very useful. Users don't know. Ignore the single E, extend them, in which direction? Okay, now this brings me to the point of introducing the concept of neural networks. Have you ever heard about neural networks, machine learning? It's one device, so neural networks, there's history of neural networks, but the way I'm introducing them now is not sort of to model neurons really, which in fact was one way in which they were portrayed in the beginning of the early 90s, but as a machine learning device. In principle, and that often is referred to in its simplicity as a perceptron, the idea is the following. You have in this blue input units and you have some output unit. You have a connection between these, a junction, J. You have a value, the connection, and the way this system operates, it takes the value 1, multiplies it by the connection value here, J11, and that would be J11 times 1 coming in here, plus J12 times 1 in this particular case would be 0, so the input side here would be 1. And then you have some sort of sigmoid trigger function, nonlinear trigger function here, that determines what comes out here. The first step is simply you multiply two vectors. Okay? The second step is essentially the sigmoid function or the nonlinear function here is a decision function. And you can perceive it as it decides is it larger or smaller than some value. So it's very, very, very negative here, you switch to 0, very, very, very positive, you switch to 1. And something in between. What you can do with that is you can do simple problems. You can classify simple problems, and the way you train, let's just assume you have two types of points here, the dark circles and the open circles, and you begin with a setting on the first stage. Then you compare the setting in the first stage to what the system was supposed to get. So in this particular case, the way it's initialized, I get a certain output, and then I know how wrong I am, because for this is supervised learning, so I know the answer in this particular case. I want to map these points to 1, say, and these points to 0. So I know the answer. I can compute whatever I have for random initialization of the junctions here. I can find what I have according to the functions that I described to you, having some sigmoid here, and I can compile the error. How do you train that system? Well, you essentially go down gradient descent, steepest descent. The simplest way, you have some sort of function of the junctions with respect to the error. You go downhill. And you avoid that you get trapped into local minima by introducing something that physicists call temperature, essentially allows you to do uphill moves. In principle, so the way this is trained, you present examples again and again, and when you present these examples every single time, you change according to the error you make and flip around with your connections, so the free variables in the game are the connections, and this way, presenting the examples again and again, you train the system to bring up the same example, or to get the correct solution. Essentially what you do is you introduce lines in space. And lines in space can classify these open circles from the closed circles, right? Now, there are different lines here, because depending on the details of the way you do the training, you may get a different solution. But in this particular case that I show you, all the lines correctly predict, right? So they are all equal. That's often referred to as the XOR problem in the literature. How would you classify those with a line? You want it? Oh, yeah. Oh.
Can introduce a new dimension that split the data.
Has everybody heard what he said? Can I ask this side of the room, have you heard what he said? Oh. Why do I repeat questions if everybody hears? So you can introduce. So one thing, can you do it with a single line? Sure. Sure you can. It's a line, right? Be careful in the way you talk. Not a straight line. Only because I show you 5 straight lines doesn't mean that every line is straight. But anyway, yes, you're right, you cannot do it in a straight line, and in fact the neural network that I described so far only introduces straight lines, so you cannot do it with the formalism that I presented so far. How now, so this type of line you can't get. You could obviously do it if you had 2 lines. And that essentially brings up the issue, how do you introduce two lines? And two lines you introduce by, in fact, increasing the dimensionality of the problem. The way it's done in a neural network, you introduce what is referred to as a hidden layer, in red here. It's hidden because that's input, that's output. This is not visible. It's just some computation that the device does in between. The way the computation is done is the same as before. You simply multiply what comes out here by that connection. That's the input. Then you have a sigmoid function deciding what is the output. And you do the same thing here. Since in this particular case it's two layers, you end up with this equation. It's simply the sum over the sum. It's very, very simple. Now, you could, if you wanted to have more dimensions, add additional layers. And again, the literature is full of people who advertise that. Very, very misleading. The only way it does, it eats up your computation time and makes you train a device that is not controllable. A much simpler way to do it is you just blow up the number of hidden units. And in principle, by doing that, you can sort of classify any kind of problem. Again, what it does, it's a vector multiplication. You project vectors upon one another and then introduce lines, hyperplanes in space. That essentially is the idea. The free variables are the connections and you move downhill. The first statement that I made, when you blow up the hidden unit, you can classify any problem. That is, and now we're back to the secondary structure issue, as long as the input that you have allows the classification of that problem. If you have the identical input vector for state blue and red, then the device cannot do it, no matter how many input units, because that is impossible. But as long as there is a way to map the problem, blowing up the hidden number will classify this problem. What's the problem that you run into when you blow up the number of hidden units? What's the possible problem? So again, yeah?
Training becomes really slow.
That's a very, have you trained a neural network?
Yes, that's right.
So I got into many fights over the course of my life because I essentially ate the CPU of various institutes in the world entirely for training the neural networks. And many people got very, very unhappy with me. I showed you, during these talks, I showed you many structures of structure biologists who couldn't do their structures in time because my neural networks were training for way too slowly and I was behaving not in a nice way. Now, that is true, that is one problem. But that's not really what I meant. There's another problem. So any kind of machine, essentially what you do by blowing up the number of hidden units, you blow up the number of connections. By blowing up the number of connections, you blow up the number of free parameters. That's why you suddenly can classify problems that with fewer free parameters you cannot classify. But what is the problem when you, any kind of machine learning device, when you blow up the number of free parameters too much?
If you give me enough parameter, I can classify everything, and that won't be a very good classifier because it will be very particular to this case.
Overtraining, learning by heart are the words. So this is exemplified here. You have the training time, you have the degree to which you do right. Black, or actually the line is green, is the training set. Blue is some other set. And you see that there is a point above which you overtrain, meaning you only learn for the examples in the training set. You optimize your many, many free parameters to the reality of that instead of that. And that brings us back to the entire idea of machine learning. Machine learning, you try to learn a problem, but exactly you try to do it because you hope that you extract principles, that you extract something that is valid, not for the training set, but for any set in the data that you would ever see. And in some sense, until this point here, these two lines indicate that you're right, that you extract information that is valid for a data set that is not the set that you use for training. How do you create these two different data sets? So let's refer to this as the training set. How do you create the other set? Anybody? You have used machine learning.
You just split all the data you have and reserve a chunk for testing, and use the others for training.
So that circle represents, and I'm not entirely sure whether I bring this example again, but I can as well bring the story here. So that circle represents all the data that you have, and the statement here is I take some fraction, I use it for training, and I have some other fraction that I use, that is the blue line. Is that all I need to do? And that, in fact, is often referred to as cross-validation. Is that all I need to do? Let's give you an example. We are talking about secondary structure prediction here, and I said that we sort of want to beat the 60% mark. So I have method A, and method A reaches 60%. Method B is trained on say I have 100 proteins, so method A is trained on 50 proteins, and method B is trained on 50 proteins, and method B trained on 50 proteins reaches 62%. And that's the training performance. For what you call testing, which is also 50 in this particular case, this one here is 58, and this one is 58. Let's not discuss about the issue when can I distinguish between two different, when is a difference significant. Let's just assume any numerical difference, any integer difference here were significant. Not true, we get back to that problem at some later point. My question to you, is method B better? Obviously, for the testing set, they're equal. But at least B does better for the training set.
We should have 3 kinds of sets. So
Okay. You get to talk at some later point. That's true what you say, but I'd like to reserve that for the next one of the next questions that I'm coming. It's not so the problem that I state is not complete, is what he says, and what he attacks is why you call that test set, and we have to get back to that. But let's stay on the problem as it's phrased on the whiteboard. Oh, yeah.
Well, I'm going to say that what we want to get out of the whole exercise is a trained machine that will classify unknown proteins properly.
Yes.
And so in this case, for the overall performance of the neural net, we should consider both the performance on the training set and performance on the testing set. And in that case, a neural net that is good on the training set and equally good on the test set is probably one that is more likely to be useful for unknown proteins than one that is worse against the training set.
I don't know whether everybody of you heard that. So essentially the argument here is I care for as many proteins that I haven't seen before as I can. In this particular example that I show, I have 50 proteins assessed here, 50 proteins here, and for those 50, I cannot tell anything about those, they are equal, but for the 50 I show here, this one is better, and they're also 50. And maybe they resemble something else that I'm going to see tomorrow. So go with method B as the rationale here. And in many ways this is impeccable. There is another point that again, it's a little bit sort of the issue of straight line that I brought up. I never told you that they use the same proteins for training and testing. Okay? It's the first thing you have to look up. Remember we had the discussion in CASP. You really have to make sure that what they used here is the same proteins for the two. I have not made any statement. All I said was 50 proteins. And they may just have taken the same data set initially, but they may have split it in different ways. One does it alphabetically, and one does it randomly. And both of those are, in many ways, you could say equal ways of doing it. That would already sort of make these numbers incomparable. So that's the first thing you have to check. These are really the same statements. The second way that you have to look at this, or the second issue that is very, very important, is you cannot just take half for training and half for testing. No matter what you do, you have to use everything that you have for testing. So in this particular case, the way to do it is you have to, in fact, set up two different networks. One trains on this half, one trains on that half. And then in the end, you come to a statement on all 100. Now, if I had made that on all 100, what about your statement now? Your statement would be valid still, right? Because there is some set for which B has a better performance here. Okay, there are now two other issues that are very crucial. I never told you anything about these proteins here. So far we have been calling this test set. Is there any additional thing any additional test I have to do?
The sets need to be unbiased.
What does it mean, unbiased?
That we cannot have some kinds of proteins in the test with another family in training, but they should be all mixed up.
So you would use the term, I cannot have a protein family that is both here and here.
We should have that.
We should have that, or we should not?
We should pick at random, basically.
We should pick at random, but that is not making the point. Picking at random does not imply at all that you look at any overlap between these sets. Your first statement convinced me that I have to somehow look at the overlap. The second statement says pick at random, but they are contradictory to each other. So I agree with the first statement. We clearly need to check out overlap. How can we check out overlap between the two sets, or any number of sets? I know you want 3, but we will have the same problem there.
I think that choosing randomly is the best way to avoid that.
Nope. It's very simple. I give you proteins that are derivatives where one amino acid change. And those are 100 proteins that I can easily give you. There's absolutely no way you can develop a secondary structure prediction method with that. And in fact, you would immediately, if you had a method that sort of removed bias appropriately, you would realize that all I gave you is one protein, and with one protein you cannot do cross-validation, or end of story. But you wouldn't realize that if you just randomly chose, without checking explicitly the overlap. How can you look at the overlap between these two sets? Sequence alignments. You just run alignment methods and you look at the degree of overlap you have. What degree of overlap is okay? Well, remember, we do comparative modeling. Comparative modeling, I told you, is a way to establish a similarity between a protein and a protein of known structure and a protein of unknown structure, and to predict the 3D structure, if they're similar enough, the 3D structure for the protein of unknown structure. If I can apply comparative modeling, I don't need a secondary structure prediction method. I can predict the entire 3D structure. So I have to make sure that the overlap between these two is such that I could not apply comparative modeling in between. Okay? If those here are a comparative modeling distance to these, everybody would just run Modeler or Swiss model, and I wouldn't care about your secondary structure prediction method and would do much, much better. So that's what you have to see here. Okay? And that, now I present a solution here for secondary structure, or I present a statement here for secondary structure. If you develop methods, this is a very crucial part. You have to consider what degree of overlap you do accept or you don't accept. It has to do with what are you up against. is your goal? What is it that you want to establish that nobody else can do, or that method to establish? And you may in different cases, so we have spent a lot of time on establishing different thresholds, different criteria for this separation. That may be very time consuming. But now let's get back to the next issue here. The next issue, and my statement, I do understand what you said, but my radical statement, and certainly in the context of making simple statements that people memorize more easily, just throw away training data whatsoever. I don't care about it. What I care about is really how it does in testing. So that brings me to the next issue. So let's assume A and B are not different methods. Method A uses 10 hidden units. Method B uses 20 hidden units. And method C uses 25 hidden units. And what I get is 58, 60, 59. So now what I publish is, and this is again, now is my test set 100 proteins, and I made sure that there is no overlap here. For instance, that I cannot align them in pairwise comparisons, that they have less than 25% sequence identity, or something like that. Tested on these 100 proteins, no overlap between training and testing set. I make the statement in my method that I have a method that is 60% accurate, and my best way of doing that is a neural network that uses 20 hidden units. Is that right? Is that okay? Kosher. Is there any mistake here? Can you attack me?
Well, it only works for your specific starting conditions. The size of your training set is all.
That's a very general attack that you do, and let's call it mean one, because you say, well, okay, you take all the proteins you have today, you use everything that has no overlap, it turns out to be 100 proteins, but prove to me that for the proteins that will be published over the next 10 years it would work. That's mean, because I absolutely cannot do that. And in fact, it's the end for any kind of prediction method, because we can never prove that it works for something that we don't know today. Unless you have, I would attack you back now, unless you have a better statement about why my set today is incomplete or what is wrong. I don't like your statement, because it takes my survival away. But let's keep these emotions out. That's not the point I meant. And in fact, I will address that issue at some later point. We can address that issue to some extent, but what is wrong here is something else. Something is wrong here that is way, way worse. Yeah?
Well, if you have more neurons, you need more training time.
That is true, but come on, we have the LRZ. They build computers and they stand around and, you know.
No, what I mean is, did you also use...
Mind you.
Did you use the same training time for all of the configurations?
No, I don't want to use the same training time. I really don't care. I want to have a tool that is going to be used millions of times by the world. In fact, this particular tool that I described has been used millions of times. And you know, whatever time it took for training it is nothing compared to all of that. So if I can do better, in this particular case, however, my main point is in terms of your CPU time here, this is 25 and this is 20. So this, in fact, is the optimum is at the point where I take less time. Was there a point?
I was asking what the numbers were, these 10, 20, 25 minutes.
Yeah, oh sorry, yeah. So that is 20 units. That's a made up example, of course.
What are they? What do they represent?
Number of hidden units, so the number of free parameters. Or it could be something else. I'm just making it up here. So essentially I have the same three methods here. It's just neural networks that train on 50 proteins, 2 times. What I show you is the testing on all 100, where every protein that I have available that is unique, unbiased, is an unbiased data set, so there's no pair in there where I can apply comparative modeling to each other. Of that, at that point, say I have 100 proteins, I have many thousands today, but let's say I have 100, and I show you the performance accuracy of 3 different neural networks. One is trained on 10 hidden units, one on 20, and one on 25. Those are the performances, so one reaches a Q3 of 3-state accuracy. How many of the residues are correctly predicted in either of those 3 states correctly? Just a sec. 3 numbers, 58 for 10 hidden units, 60 for 20 hidden units, 25 hidden units give 59. I conclude in my publication that in fact the best method is 60% accurate and it uses 20 hidden units. And I'm asking, is the logic okay?
Shouldn't we try the methods of the same number of hidden units?
That's a little bit what he says. But the same number of hidden units, in some sense he says something similar, because he says you should also put into account how long it takes training. So put other values in. But listen, I have a machine learning device. I have some free parameters that I use. In this particular case, it's the connections. The connections is done by the number of hidden units. If I cannot classify my problem with 10 hidden units because it's too complex, I need to move to 20. Why should you demand of me to use 10? Because the power of the system here, the machine learning device, is in fact, all I need to do is blow up the hidden level and I can handle a higher level of complexity. That's the power of it. Let's not restrict that power. Again, the problem of overtraining comes with it. We have discussed that. We have solved that. But given that we have solved in some sense, there's something else in here, but the trivial overtraining, so it's 1, 2, the trivial overtraining is taken care of. So then give the power to the machine, is what I'm saying. So no, it doesn't have to be 10 for both. You were the first and then?
Yes, I have a similar argument to the ones that were expressed, but if we have more free parameters, wouldn't we need more data in order to have an appropriate
so that may be true, but I don't have more data. So now let's get back to this question. Maybe what we don't see here is the question, is my data enough for that number of hidden parameters that I use? So maybe I blow it up too much, but this is my testing value. This is not my training value. So if the testing value goes up, you may argue this is because we are still not overtraining. I still have a system that operates below the overtraining point. As long as it operates below the overtraining point, you're not overtraining, by definition. Yeah?
You might be undertraining, you might not be training.
Undertraining, you may be, yes. But that is shooting yourself.
If you're saying one method is better, or you haven't given that method what it needs to be good. So I think that's the but this, we're not in Germany here. We're not trying to make everything optimally right. So I'm publishing one method. My question is, I'm not saying this is the best you can ever do. I'm saying I have a method that is 60% accurate. Now, can you attack that? It's not, I mean, come on, don't blame me that I don't say it's 61 because I could have done something else and it would have been 61. That's not my mistake. I'm limited. I'm just human.
I have a more general remark, but if we assume that we want to classify 3 types of protein, A, B, or we have 3 types, A, B, C, maybe the method A is very good on protein A and not that good on B.
That's true. So there's more to be done. But again, that is not, you're bringing up issues that are on the level of the next, so you all bring up issues that we will discuss again. But here's something very, very wrong. And I thought that you would say it. So what did you mean by your 3 sets then? No, if you don't see that now.
The 3 sets before?
Yeah.
I was thinking of k-fold cross validation, and so We can use two sets. So if we split our first our data in two, with one validation set that we won't touch anymore. And then for the testing set, we can split it into a set and testing set. And
okay, let's yes, you're right on, and I don't see why you don't see then see the problem. This is the solution, and this is the problem. So the problem here is that if I do this, I use a parameter and I look at the test set to optimize a parameter. The number of hidden units is another free parameter. It's true, the major free parameter in the game are the connections here. But the number of hidden units is another free parameter. And in my publication, I would use that test set that I should pretend to have never seen before to optimize a parameter. And that's wrong. That's absolutely wrong. I should never do that. I can do that on a data set that, in fact, let's call it cross-training. And that in this case has another 3 proteins, and then here's the test set that has another 33. So in the case where I have training set and then there's the cross-training set, this is here when I should choose my parameter, stop training or whatever I have to do, and then run my test set and not change anything. So if that is my cross-training set, then I should in fact choose the 20 hidden units. And then assess how well this method works for the test set and not change anything anymore. It may very well be true that for the 25, for the testing set, it would have been better. But that's the end of the story. And then I in fact have to publish whatever it may be, that for the test set, what is for the cross-training here 60, that for the test set, the numbers look like 57, 57 and 58, and that in fact rather than using this set here I should have used that one. But that's the end of the story, and I have to publish that value. That's why I need 3 sets, and that's why this is a complication. Never produce different results in your paper, and choose what I call the cross-training here to choose what of the methods is best. And we all see that. So almost every paper or publication has something of that type. Makes some mistake of this type. That you don't know which algorithm is best. Is it a neural network? Is it SVM? Is it something? So you say that for my test set, the 50 proteins, the SVM turns out to be better, so I publish the paper saying SVM works better. Again, as long as you do that on that set, it's fine. If you do it on the set on which you publish, you made a mistake. And that very, very often goes very, very dramatically wrong. Now, let me just complete. We will not get through today's lecture. I'll leave it to next week. Complete one point here. This is the idea of overtraining, but here's a real case. Case for secondary structure prediction. That, in fact, is training. That is this testing, or cross-training. So you see there is some sort of optimum. There is some sort of effect of overtraining here, somewhere here, because it never gets up to the same point. But it's nowhere near as dramatic as it always in the textbooks. And reality is much more similar to this one than to the way you see it in textbooks. So the result of that is not always easy to find the point of overtraining. You have to be very careful, is what the statement here is. With that then, we will return to secondary structure prediction and essentially ask the question whether applying a neural network to the problem of secondary structure prediction will solve these problems, that we don't get to 100%, that we have low beta strand, and that we have short segments. And that we will do on Tuesday. Thanks for your attention.